In this presentation, we will be focusing on the gene editing technology called CRISPR-Cas9. In order to understand anything relating to gene technology, we must first introduce basic genetics. Then we will go through the mechanism, and finally, we will explain uses and the impacts of the technology in society. Obviously, when discussing genetics, DNA will always be talked about. DNA is nicknamed the code of life because without it, life would cease to exist. DNA is the genetic information that makes organisms look the way they look and function the way they function. We will discuss later how DNA actually provides the information necessary to do this, but for now, let's talk more about the structure of DNA. DNA is a double-stranded helix composed of a deoxyribose sugar and a phosphate backbone. It contains alternating nitrogenous bases that allow genetic information to be replicated, genes to be expressed, and information to be passed down. These sequences of DNA are organized into chromosomes within the nucleus of cells, and mitosis and meiosis allow for these chromosomes to be inherited from generation to generation, but that's a topic for another day. So how do alterations in DNA affect an organism's functions? The central dogma of genetics is DNA to RNA to protein. Through transcription and translation, DNA is read to create a specific protein with a certain function. Therefore, the protein that is created corresponds to the original sequence of DNA that was copied. Certain sequences contain information for specific proteins, and if a sequence has a mutation, the protein translated could change. Proteins do nearly everything for us, including energy production, transport, immune response, and much more. The structure of a protein dictates its function, so if the shape is altered by a mutation, the protein's function can be altered. As a result, an organism's phenotype can change if a necessary protein cannot function with its altered shape. So how does this all relate to gene editing technology? Well, the basis of gene editing is to restore the function of a mutated gene. As we've discussed, if the DNA sequence of an organism is mutated, the protein that it encodes for can lose or change its function. So what gene editing technology does is it locates this mutated gene and modifies the sequence to restore the function of that protein. As a result, the protein would function normally, essentially eliminating the negative consequences that mutation had on the individual. This technology is extremely powerful, and with more time and research, could possibly be used to cure many more genetic different diseases. So CRISPR-Cas9 stands for clustered regularly interspaced short palindromic repeat, which is basically a fancy way of saying a short partially repeated sequence of DNA that is found in prokaryotes. These repeats were initially discovered in E. coli in the late 1980s. However, the function of these repeats weren't discovered until more than a decade later. It was discovered that CRISPR and its associated protein Cas9 were naturally occurring endonucleases, or basically genetic scissors for DNA, that helped strengthen the immune system of prokaryotes. To defend themselves against viruses and bacteriophages, they used these short repeated sequences and the Cas gene located close to them to cleave nucleotides. In 2012, it was discovered that CRISPR-Cas9 can be used to alter any DNA sequence if the right template was provided. As a result, CRISPR-Cas9 is considered to be the most efficient and effective gene editing technology we have discovered so far. So there are two different types of CRISPR-Cas9 systems that are distinguishable based on the structure and the function of the Cas proteins associated with them. Class 1 systems consist of a multi-subunit protein complex, and Cas2 systems consist of a single Cas protein. The type 2 complex, which is part of the Class 2 system, is the most used and studied because the structure of the CRISPR is pretty simple. This complex is composed of two main components, guide RNAs or gRNAs, and CRISPR-associated Cas9 proteins. The first Cas9 protein used was extracted from the bacteria that causes strep throat. In a large DNA endonuclease domain that is responsible for creating a double-stranded break in the targeted region of DNA. This endonuclease can be thought of as a genetic scissor that cuts DNA at specific sites. This domain is comprised of two regions called the recognition or rec lobe and the nuclease or nuke lobe. The rec lobe is responsible for binding gRNA, and the nuke lobe is responsible for cutting the DNA and initiating the binding to the target DNA. gRNA in the CRISPR-Cas9 system has two essential components, CRISPR RNA or cRNA and transactivating CRISPR RNA or tracer RNA. The cRNA, typically about 18 to 20 base pairs in length, plays a crucial role in specifying the target DNA by forming base pairs with the corresponding target sequence. On the other hand, tracer RNA consists of a lengthy sequence of loops, serving as a binding scaffold for the Cas9 nuclease. However, in gene editing, In prokaryotes, gene RNA is employed to target viral DNA as part of their immune system. However, in gene editing, crRNA and tracer RNA can be combined to form a single guide RNA, or an sgRNA. This fusion enables the targeting of virtually any gene sequence that is intended for modification. So the mechanism of CRISPR-Cas9 can be divided into three main parts: recognition, cleavage, and repair. The first part, recognition, happens as the Cas9 protein, when bound to its guide RNA, initiates a search for complementary target DNA sites, requiring both base pairing between the 20 nucleotide spacer sequence and a protospacer in the target DNA, and the presence of a conserved protospacer adjacent motif, or a PAM sequence, adjacent to the target site. The PAM sequence is crucial for distinguishing between self and non-self sequences, and mutations in the PAM can disrupt Cas9 cleavage activity. Cas9 initiates the search by probing for a proper Cas9 initiates the search by probing for a proper PAM sequence before looking at the flanking DNA for potential guide RNA that is complementary. Once a target site with the proper PAM is found, Cas9 triggers local DNA melting and RNA strand invasion to form an RNA-DNA hybrid, or an R loop. After PAM recognition, Cas9 induces local DNA melting and forms this R loop. The unbound target DNA strand forms a pseudo-A form RNA-DNA hybrid. Cas9 bends the DNA helix, creating a global bend and preventing rehybridization, or in other words, the R loop collapsing. The next step of this process is cleavage. Cas9 is activated for DNA cleavage. The H&H and RUVC nuclease domains cleave the target DNA, producing a predominantly blunt-ended double strand. The conformational state of the H&H domain, driven by RNA-DNA hybridization, controls RUVC activity for accurate target DNA cleavage. The non-target DNA strand plays a key role in H&H repositioning, stabilizing the H&H nucleus domain in an active conformation. Cas9 cleaves a partial target duplex nearly as efficiently as a fully paired DNA target, highlighting the importance of the non-target strand. The interaction between H&H and HEL2 formed upon bonding to DNA effectively locks the H&H domain in an activated conformation for DNA cleavage, resulting in double-stranded breaks. The third step is repair. Most cells already have processes involved in repairing double-stranded breaks, or DSBs. As a result, these processes are used to repair the cleavage induced by CRISPR-Cas9. There are two common mechanisms that are used to repair these DSBs, which include non-homologous end joining, NHEJs, and homology-directed repair, or HDR. NHEJ is a mechanism present in all stages of the cell cycle. However, it is very prone to errors, and as there is no template DNA, and the broken strands are simply just fused back together. For instance, oftentimes this process will result in an insertion or a deletion at the cleavage site, leading to a frameshift mutation. This shift can create a premature stop codon or change the sequence of amino acids in the protein, creating a non-functional gene or a knockout. On the other hand, HDR requires a homologous DNA template, but it is a precise way to insert a new fragment of DNA into the cleavage site, creating a knock-in. For CRISPR-Cas9, large amount of these homologous DNA fragments are needed to perform a precise insertion or replacement at these DSBs. This process helps eliminate the errors produced during NHEJ. Thanks to the processes that already exist within cells, introducing new DNA into genomes is theoretically fairly simple. However, we still need more time and research to perfect this gene editing technology.
Today is being used as a potential therapeutic for many different types of diseases. Since this technology is fairly new, many of these cures are still in their beginning phases of research and clinical trials. One of the most recent CRISPR-Cas9-based therapeutics is a cure for sickle cell anemia. An individual has sickle cell anemia when they have two recessive alleles for the hemoglobin gene. Hemoglobin is a protein that carries oxygen in red blood cells, and an abnormality leads to changes in blood cell structure. These abnormal blood cells can also be sticky, which would prevent blood from flowing properly throughout the body, which not only damages organs, but can also affect the heart by causing strokes and other heart diseases. The blockage of blood vessels in sickle cell anemia patients is known as vaso-occlusive crisis, or VOC. Currently, there are medications to treat the disease. Stem cell transplants are another cure that scientists are working on. In this process, patients undergo chemotherapy to remove the stem cells in their bone marrow to later replace it with stem cells from a donor. The problem with this is finding a donor that is compatible with an individual's specific genetics. For this reason, a recent gene therapy called exa-cel that is being reviewed by the FDA uses CRISPR-Cas9 instead. Researchers have found a genetic alteration in individuals that is not harmful and causes them to continue producing fetal hemoglobin instead of adult hemoglobin. Exa-cel uses this information and uses CRISPR-Cas9 to disable the gene that turns off fetal hemoglobin production, B-cell lymphoma 11A or BCL11A. This is beneficial since the fetal hemoglobin has the correct structure and functionality to ensure proper blood flow. This therapy collects the patient's cells to edit and then infuses them back. Recent clinical trials have been very successful and showed a decrease in VOC symptoms. The final FDA decision about this therapeutic is going to be made by December 8th. CRISPR-Cas9 has even been used to study and potentially prevent the spread of cancer as well. The first gene therapy to enter human trials and use CRISPR was for refractory lung cancer. This therapy deletes genes that prevent cancer fighting in extracted T cells. When these cells are infused back into the patients, they are able to target cancer cells through their better antigen detection. The results from these trials are now being used to create treatments for other cancers and diseases as well. Treatments for bacterial and viral infections using CRISPR-Cas9 technology are also highly researched, especially since they come from the same roots. For example, in 2017, as the HIV and AIDS epidemic continued growing, researchers found a CRISPR-Cas9-based treatment that prevents the replication of the virus and eliminates it from infected cells. This gene editing technology has even been used for multiple reproductive medical practices. CRISPR-Cas9 can help prevent genetic diseases from being passed on to offspring by editing the DNA of an individual's germline cells. This sparked many ethical debates regarding gene editing's impact on society. One of the biggest issues regarding CRISPR-Cas9 and gene editing technology in general is the ethical concern in regards to the genetically modified individual. In addition to this, there's a concern that by removing a deadly disease, there is the high risk of introducing a different harmful mutation. For this reason, many find it unsafe to modify an individual's gene, and especially unethical to alter an embryo's DNA even before having the opportunity to give consent. Gene editing can even reduce diversity in the human population. By introducing alterations in the germline cells, the DNA that is passed on to future generations is therefore modified, affecting the gene pool of the whole population. Diversity in the gene pool allows for a population to grow, since the differences from individual to individual gives an advantage to some to better adapt to the changing environment that can further be passed on to offspring to better fit in this world. Another problem with CRISPR-Cas9 technology is how expensive it is. Since it's still in its early stages of development, it costs a lot of money to buy and implement. For this reason, those who are wealthy in our society will continue to benefit, while the poor are left at a disadvantage. With that being said, when an individual has an abundance of money and wants to use it to genetically modify how their child will look, where is the line between using scientific technology to remove a harmful disease versus playing God to pick your child's eye color or hair color? The CRISPR-Cas9 technology has been the topic of many ethical debates. It has led to key developments in many aspects of the scientific field. In regards to research, CRISPR-Cas9 allows scientists to manipulate genes to study expression levels using knockout and knock-in processes. As discussed, CRISPR-Cas9 is currently also being tested as a therapeutic for many diseases. This allows for a major development in medicine since many individuals can be healed or are relieved from stresses of certain diseases. For individuals that know they will pass on harmful genetic disorders, CRISPR-Cas9 helps eliminate the high probability of this occurring by altering embryonic DNA. Overall, CRISPR-Cas9 is one of the most researched gene editing technologies used today and has the potential to become the cure for many diseases such as cancer. Thank you. Here are our references.